Really my pleasure to introduce Dr. Scott Turner, PhD, MD, and I want to thank you for joining us today live from the St. Boniface Hospital Research Center here in Winnipeg. Dr. Turner is the Vice Chair for Clinical Research and Professor in the Department of Neurology and Director of the Memory Disorders Program at Georgetown University in Washington, D.C. He received his doctorate in medical degrees from Emory University in Atlanta, and that was followed by an internship and a residency in neurology in a Howard Hughes Medical Institute funded fellowship in behavioral neurology at UPenn in Philadelphia. He's board certified in both psychiatry and neurology, and prior to moving to Georgetown, he was the chief of neurology service at the VA hospital at Ann Arbor Healthcare System in Ann Arbor. And he was also an associate professor and associate chair in neurology at the University of Michigan in Ann Arbor. And that's actually where I first met Scott while I was working there at Warner Lambert that transitioned to Pfizer. So Dr. Turner now directs the Memory Disorders Program at the MedStar Georgetown University Hospital. His research is focused on developing new treatments and biomarkers for neurodegenerative diseases of aging, particularly mild cognitive impairment and dementia due to Alzheimer's disease. Dr. Turner is also on the steering committees of the Alzheimer's Disease Cooperative Study and the Alzheimer's Therapeutic Research Institute. And he's also a site PI for the NIH-funded Alzheimer's Clinical Trials Consortium. And I guess trial-ready cohort for the preclinical prodromal Alzheimer's disease. So I hope I got all that correct.
That was great.
He's also PI or site PI a number of different multi-center randomized placebo-controlled trials, including resveratrol for Alzheimer's and nilotinib also being tested in for Alzheimer's. But I guess you folks in your team have done work with nilotinib in other diseases like Lewy body dementia and Parkinson's there at Georgetown too. And of course, nilotinib is a compound we just recently collaborated on with Scott, and that paper was just released very recently in the journal Aging and Disease. And if you are interested in Dr. Turner's work and the Memory Disorders Program at Georgetown, please see memory.georgetown.edu. And the title of his talk today, you can see on the screen, is Aging a Target for Alzheimer's Disease Drug Development, a Tale of Two Trials, Resveratrol and Nilotinib. So welcome, Dr. Turner.
Thank you. Thank you for the invitation. I'm just going to talk a little background on Alzheimer's and introduce the biomarkers because we use these in the phase 2 clinical trials that I'm going to talk about, resveratrol and nilotinib. So, this should be a review for everyone, but basically these are the amyloid plaques and the neurofibrillary tangles on a silver-stained brain section, which is derived from this A-beta peptide, which changes conformation to insoluble forms and aggregates and precipitates in the brain. It comes from APP, the precursor protein, amyloid precursor protein cut by beta and gamma secretases to release A-beta. And this is a normal process. It happens in all of our tissues, including our brain. Most APP is cleaved by alpha secretase, releasing this benign fragment, P3, which causes no problems. But as we age, the clearance of A-beta slows, and then it begins to accumulate and is neurotoxic and causes problems. So one area where we have made a lot of progress in Alzheimer's in the last 20 years has been genetics. So there are 3 autosomal dominant mutations in presenilin 1, presenilin 2, and APP that cause Alzheimer's at an early age. And there are more than 100 different variants found by GWAS studies that influence Alzheimer risk. Not much of an influence compared to these autosomal dominant mutations. And then there are these 2 genes sort of in between. One of the
funding is needed, how it fits in the overall research program, and how funds are distinct from the funds currently held. For EPRs with no previous
Could somebody,
I'm sorry, there's a background noise. If you could turn your microphones off in the background.
Could someone mute their, yep, okay. So here's the APOE4 variant. Carrying 1 copy increases your risk of Alzheimer's 3 times, 2 copies 8 times. And then more recently, there's a TREM2 variant. This is a triggering receptor on myeloid cells, which is carried by microglia. And a variant influences your risk of Alzheimer's approximately 3 times. So this clearly indicates that there's a lot of immune-mediated pathologies and inflammation as part of the disease, Alzheimer's disease. We're not quite sure how ApoE is working, but one mechanism that ApoE4 increases risk of Alzheimer's is that it increases your risk of having a positive amyloid PET scan. So here are the 6 genotypes and the approximate population frequency of the 6 genotypes. You'll notice if you have ApoE22, there's no amyloid deposition with aging, and if you have 4,4, there's this markedly increased amyloid deposition with aging, and then the other genotypes are in between. We're not quite sure how this works or what the mechanism may be, but clearly ApoE4 somehow influences amyloid deposition to promote Alzheimer's disease. And as I said, there are now more than 100 different genes. Here are the 3 autosomal dominant mutations. Here's ApoE, here's TREM2. More than 100 different genetic variants are now influencing risk of Alzheimer's disease. One way to sort of look at this or try to make sense of all these genes is to classify them into their function. So here is the early onset familial Alzheimer genes. All of these promote A beta formation from APP. A lot of these genes are involved in endocytosis and cellular and protein trafficking. And we'll come back to this, but some of them are also involved in innate immune and brain inflammatory response genes. So clearly these processes are implicated in the Alzheimer's pathologic process. This is the famous cascade, amyloid cascade hypothesis. And we start with APP turnover going to A beta accumulation, which is neurotoxic, promotes tangle formation, you develop mild cognitive impairment, astrocytosis, microgliosis, inflammation. This sets up a focal encephalopathy, which is detectable by slowing on EEG, for example, or hypometabolism on a glucose PET scan. This leads to loss of neurons and transmitters and synapses, leading to atrophy, white matter loss, dementia, and death. This takes 10 to 20 to 30 years to happen. And here are our causes on the left. Number 1 is aging. And then ApoE4, Downs, and having one of these familial Alzheimer mutations, which is only found in about 100 pedigrees in the world. These are when the biomarkers become abnormal, and I'll show you what these look like. And these are our current treatments, 3 cholinesterase inhibitors and a NMDA receptor antagonist. You can see that they're pretty downstream, so they only have like modest benefits, and we don't really think they're disease modifying in any way. You can also look at all the different biomarkers and see when they become abnormal. So we classically studied dementia patients, and now we're becoming more interested in mild cognitive impairment, which is the precursor stage to dementia, several years in the MCI state before converting to dementia. And now, because of these biomarkers, that is another success story in Alzheimer's in the last 20 years, we've defined and discovered this preclinical state when the biomarkers are becoming abnormal, and yet someone is still cognitively normal. So these are cognitively normal individuals with preclinical Alzheimer's. This is when memory and cognition starts to decline, and this is when function starts to decline as well. So these are our 3 major classification schemes. And there's evidence for and against the amyloid hypothesis on this figure. One is that the first biomarker to become abnormal is amyloid deposition in the brain, or decreased CSF A beta levels if you do spinal tap. So the fact that this is occurring first sort of promotes supports the amyloid hypothesis, because this is the first pathology we see. But you also see that there's this ceiling effect, and you cannot really predict someone's cognitive status from their amyloid burden. So someone with a high amyloid could be normal, could be MCI, or could be Alzheimer's. And so there's a disconnect between the amyloid burden and the cognitive status. And this argues against the amyloid hypothesis. This would suggest that there's something else that's driving cognitive decline. To help organize these biomarkers, we've developed this ATN framework. So A is amyloid, T is tau or tangles, and N is neurodegeneration. So this is amyloid PET scan and CSF levels of A beta 40, 42, or even better is their ratio. And now more recently, plasma biomarkers. And T is tau, which is tau PET scans, CSF and plasma, phospho-tau in particular, phospho-tau 181 and now phospho-tau 217 and other phospho-tau epitopes. And then the N is neurodegeneration detectable by volume loss on volumetric MRI, hypometabolism on glucose PET, or CSF total tau or neurofilament light and other proteins. And the people with Alzheimer's disease are either A plus, T plus, N plus, or A plus, T plus, N minus. So here we go from if you're cognitively normal, your preclinical Alzheimer's. If you're mild cognitive impairment, it's MCI with biomarker support of the diagnosis. And then this is the true classic full-blown Alzheimer's disease with dementia with all biomarkers positive. And then normal people are, of course, normal cognition and all biomarkers negative. And then there's all possible states in between. This is what the amyloid PET scan looks like. This is a PIB amyloid ligand, which was the first amyloid ligand developed. You can see medial temporal hippocampal amyloid deposition and then more diffuse amyloid plaque deposition as you go from normal to Alzheimer's disease. And then more recently, the tau PET ligands are also indicating medial temporal lobe, hippocampus, entorhinal cortex uptake of the tau PET ligand, and then spread outside the medial temporal region to other cortical regions as you go from normal to Alzheimer's. In the CSF, a low A beta and a high tau is associated with Alzheimer's. And a high A beta and a low tau is associated with normal individuals. So this is a clinically available diagnostic test performed on spinal fluid that can help support a diagnosis of Alzheimer's. And then there's also volumetric MRI, and this is another example of N. So this one, so here's an example of an A biomarker, T biomarker, A and T biomarkers, N biomarker. This is loss of volume by annual MRI PET scans. So here you can quantify the volume loss about 3% per year once you get to Alzheimer's disease, particularly in these inferior temporal regions, with sparing of the primary sensory motor and occipital and auditory cortex. And then glucose PET scan is another useful quantitative biomarker. Here's mild cognitive impairment in Alzheimer's patients, and everything in yellow and red is hypometabolic. So this cortex is not taking up glucose. This is also a clinically available diagnostic study to help distinguish frontotemporal dementia from Alzheimer's, for example. So this is an abnormal glucose PET scan with temporal parietal bilateral hypometabolism. And the posterior cingulate cortex is the part of the brain that is the most sensitive, the earliest and most severely affected in Alzheimer's disease. So if you see this signal, this is also diagnostic of Alzheimer's. And you can begin to see these changes in mild cognitive impairment as well. So another success story in Alzheimer's research has been this development of these biomarkers, and starting with the glucose PET in 1980, volumetric MRI, amyloid PET, tau PET, and then CSF proteomics, A beta, tau, phospho-tau, and now other phospho-tau epitopes. And then more recently, blood biomarkers or plasma biomarkers has really come on quickly in the last few years, particularly, for example, phospho-tau 217 is detectable in plasma, and really a breakthrough because we would really like to have a blood test for Alzheimer's disease, because obviously the PET scans are expensive and the spinal fluid tests are invasive. So if we had a good blood test for Alzheimer's, this would really help with our screening, diagnosis, prognosis, and testing drug outcome, clinical trial outcomes. This is this A beta 42 to 40 ratio is our first commercially available diagnostic blood test that's diagnostic for Alzheimer's disease or supportive of a diagnosis of Alzheimer's disease. And this only became available last year. So these blood biomarkers has been another big recent breakthrough.
Among you.
So you can see some of the trends in our Alzheimer's research. We've developed these biomarkers, glucose PET, amyloid PET, tau PET, and there are others that are in development. For example, looking at synaptic density or neuroinflammation. These are much further down in the clinical development, but may also be useful in the future. Develop the CSF and plasma proteomics, for example, A beta 42 and 40 or their ratio, phospho-tau and neurofilament light and other proteins. In general, we sort of shifted towards earlier in the Alzheimer's spectrum with our clinical trials. Our classic trials enrolled dementia patients. We're now becoming more and more interested in MCI patients and even these preclinical Alzheimer patients who are normal but have abnormal biomarkers. So we're doing more and more trials enrolling mild cognitive impairment individuals. And now these biomarkers have enabled us to start prevention trials. So we're enrolling high-risk individuals, older individuals who may be ApoE4 positive or amyloid PET positive for a prevention trial. And of course, there's been a lot of work in non-drug strategies, and there are certain lifestyle interventions such as exercise, Mediterranean diet, avoiding smoking and other risk factors, maintaining ideal body weight throughout one's life. All these lifestyle interventions are also being examined, and probably their combination is the most powerful or effective. And it's possible that one may delay the onset or prevent Alzheimer's by 5 to 10 years by doing all the right lifestyle interventions. Now, to find these individuals in the earlier part of the Alzheimer's spectrum, we've had to go to web-based screening and recruitment. So these people are not necessarily coming to our clinic the way the classic dementia patients would for our classic dementia trials. So we have to go out and find innovative ways to try to identify MCI individuals and preclinical Alzheimer individuals who may be interested and eligible for prevention trials and clinical research. And there's been a huge increase in NIH funding. I think everyone's finally recognizing the huge burden of Alzheimer's disease, not just the medical burden, but the financial burden, societal burden. And so NIH funding has quadrupled in the last few years and is really helping to develop the field. I'll just briefly mention the current drugs for Alzheimer's. I'm not going to talk much about them. These are mainly to point out that our last new drug in the United States was FDA approved in 2003. So that means that all of the phase 3 clinical trials that have been done since 2003 have not shown efficacy or safety, have not worked, basically. And so the question is, why have we not been able to come up with a new drug or a new medication for Alzheimer's since 2003? Is the amyloid hypothesis wrong, or do we not really understand what's causing this disease? And I'll just mention the leading approach currently, where most of the investment, particularly of big pharma, has been in developing anti-amyloid monoclonal antibodies to promote the clearance of A beta amyloid from the brain. And here are some candidates: aducanumab, gantenerumab, donanemab, lecanemab, which was called BAN2401, and solanezumab. So all of these antibodies have somewhat different epitopes, whether they identify the fibrils or the oligomers of amyloid. There's a difference in whether they're entirely human in sequence or human mouse chimera. And some are given IV, one is given subcutaneously, mostly once a month, but sometimes every 2 weeks. And so their dosage, epitope, affinity, all slightly differ in some way, and we really don't know what the ideal target for these monoclonal antibodies is yet until we have a successful trial. But aducanumab in particular has a very interesting story of one positive phase 3 clinical trial and one negative phase 3 clinical trial, and is now under review at the FDA for possible approval. And a decision will have to be made by the 7th of June. I don't really know what that decision is going to be. I think we probably need more information, another trial, to see if aducanumab is really effective. All of these antibodies are very effective at removing amyloid, as demonstrated by amyloid PET scan. So there's definitely a positive biomarker outcome. What's been harder to show is a clinical benefit of removing the amyloid. Some of the antibodies do slow the rate of cognitive and functional decline, but the patients are still declining. So this would argue against the amyloid hypothesis, because now we've removed the amyloid from the brain and the patients are still going downhill. They're just not going downhill as quickly as the placebo group. And if the studies demonstrate a 20% difference or better between placebo and antibody treatment, that's enough difference to warrant approval of these antibodies. Some of the studies also looked at tau tangle biomarkers, and removing the amyloid also had a benefit in some of the tau PET, for example, and phospho-tau levels in spinal fluid. So the fact that an anti-amyloid agent would have benefits on tau is supportive of the amyloid hypothesis. But again, the patients are still going downhill, even with removing the amyloid by PET scan and having that positive benefits on the tau. So something else must be driving the cognitive decline at this state. The major side effect of these antibodies is something called ARIA-E and ARIA-H, which is amyloid-related imaging abnormality edema and amyloid-related imaging abnormality hemorrhagic, which is very common, although mostly asymptomatic and only detectable by MRI scan. But it is somewhat of a dose-limiting side effect because the higher the dose, the more the risk of the ARIA-E and ARIA-H. If it is symptomatic, it presents as a headache and sometimes more confusion. ARIA-H is usually microhemorrhages, which are subclinical, but it can be larger hemorrhages, such as a hemorrhagic stroke. So there have been some pretty catastrophic events of ARIA-E and ARIA-H, where of course the drug had to be discontinued. But most of it is asymptomatic and resolves over 1, 2, or 3 months, as demonstrated by repeated MRI scans. So now the question is, are these antibodies going to be the treatment, the disease-modifying treatment of the future, or are there other strategies? And so this brings me to looking at disease mechanisms associated with aging. We know that aging is the major risk factor for Alzheimer's disease, but what do we mean by aging, and what exactly is the target? What is the molecular target in aging? So these are different sort of domains of aging, where different regions become different physiologic functions, and molecular targets and pathways become abnormal with aging. And here's mitochondrial dysfunction, impaired molecular waste disposal, so protein aggregates accumulate and become neurotoxic, impaired DNA repair, aberrant neuronal activity, stem cell exhaustion, glial cell activation and inflammation, so abnormal inflammatory responses, impaired adaptive stress responses, dysregulated calcium homeostasis, oxidative damage. Is there one particular driver of pathology in Alzheimer's disease, or are all of these functioning? Another one is cell senescence, which is very apparent in other tissues, but not quite clear what the role is in the brain. And then, of course, telomere shortening is another strategy, but again, not quite clear if this is happening in the brain and if this is a potential strategy. You can see the biggest physiologic system is dysregulation of energy metabolism. And what this is something that they may all have in common. So there's some very powerful or definite links, molecular links between energy metabolism and aging, however you want to define aging. Here's an example of environment affecting aging. So here's a truck driver who had sun exposure on the left side of his face for 28 years, but no sun exposure on the right. So you can say this sun caused accelerated aging of the skin on the left face, or you can say that protection from the sun slowed aging on the right side of the face. It's sort of a semantic issue. But there's another interesting environmental regulator of aging, and that is caloric restriction. Again, getting to that energy balance link with aging. So here is a study done at the NIH on 27-year-old monkeys, and these monkeys had 20 years of caloric restriction. On the right, these monkeys are the same age, but the one on the right had caloric restriction and the one on the left had ad lib food, as most laboratory animals are given. And so caloric restriction means that this monkey only consumed about 2/3 of the calories of this monkey, but was not deprived in any of the vitamins or anything else. And just about every outcome measure that you look at in this monkey demonstrates a slowing of aging. including activity, muscle tone. Just look at the hair and other features of this monkey compared to this monkey. So caloric restriction is a very interesting environmental regulator of aging. And the question is, what is the molecular mechanism of how this works? We don't really know if this works in humans. There haven't been a lot of studies of caloric restriction with humans. It's kind of a difficult thing to do with humans, but clearly has been demonstrated in many laboratory animals and going all the way down to yeast. So this brings me to the resveratrol for Alzheimer trial, which was we published in 2015 and also did some additional analyses on the spinal fluid and published them in 2017. And this is starting with the caloric restriction, which regulates NAD and NADH ratio. This ratio activates a group of genes or proteins called sirtuins. And sirtuins regulate acetylation of histone, so it's a form of epigenetic regulation of gene expression. So this is a molecular pathway linking energy balance to regulation of gene transcription and translation. So the question is there something pharmacologic that could activate sirtuins and skip the caloric restriction? And in yeast, it was found that certain polyphenols can activate sirtuin. One of these polyphenols was resveratrol. So we launched a clinical trial of resveratrol, and we screened 179. We ended up with 64 in the resveratrol arm and 55 in the placebo arm. This was a double-blind, randomized clinical trial. We synthesized our own resveratrol to make sure that it was pure, a pharmacologically pure agent compared to what are the commercial resveratrol preparations that are out there, where we don't really know what they are. So we synthesized and made our own pills of placebo, matching placebo or resveratrol in this particular study. We only manufactured enough for the trial. And then looked at CSF A-beta levels. And you can see that the resveratrol treated group had relatively stable A-beta levels in plasma and CSF compared to the placebo, where you see the expected rate of decline. As I said before, lower levels of A-beta is in the wrong direction. So higher levels is in the right direction. So it looks like resveratrol did have an effect on CSF A-beta levels. Obviously, we don't really fully know the mechanism. We don't know if sirtuins are activated or not. But my worry was that these lines were going to overlap and resveratrol would have no effect whatsoever. But that's not what we found. So there was an effect of resveratrol treatment in CSF A-beta levels in this small phase 2 clinical trial. In general, we found more effects in ApoE4 carriers compared to the non-carriers. CSF A-beta level 40 levels were significant in both groups and the entire group, but other plasma A-beta 40 and brain volume, ventricular volume were only significant in the ApoE4 carrier group and not significant in the non-carrier group. We also did some pharmacokinetics of resveratrol. Resveratrol is taken up from the gut and very quickly metabolized by the liver into glucuronidated and sulfonated forms of resveratrol, which of course impede blood-brain barrier transport of these compounds. And so we looked at the concentration of resveratrol in plasma, and we're talking about nanomolar levels, 13 nanograms per ml of resveratrol in plasma, and much lower levels in spinal fluid. So even these low concentrations, nanomolar levels of resveratrol in spinal fluid are having these effects on A-beta levels and brain volume. We also detected a small amount of the metabolites in the spinal fluid as well. And there's some evidence that the metabolites may also have some activity in vivo. As far as the side effects, the only side effect that was significant was in this investigations category, which includes weight loss. So those who got resveratrol lost more weight than those who got placebo. And then the only other significant difference was that there are much fewer cancers in those given resveratrol compared to placebo. And I should mention that most of the interest and funding for resveratrol has come from the National Cancer Institute as a cancer prevention agent. So we also found less cancers in our trial, even though these are, of course, small numbers. So we found that high-dose oral resveratrol, and I should mention that the dose started at 500 milligram once a day, and then increased every 3 months by 500 milligram. So that the last 3 months, people were taking 1 gram twice a day, which is a huge amount of resveratrol. But this huge amount was safe and well tolerated in individuals with mild to moderate Alzheimer's. The major side effect were GI, although this was not significantly different from placebo. There were more GI symptoms, weight loss, no serious adverse events. And the res can, and its metabolites can penetrate the blood-brain barrier to have CNS effects, can alter CSF and plasma A-beta 40 levels, and increased brain volume loss. So I think this represents less swelling or edema or inflammation. So based on this, we would like to do additional studies of resveratrol, and we're now working with a company in Jupiter, Florida that has a patented formulation that has much improved PK by forming lipidated nanoparticles of resveratrol to improve metabolism and require less oral dosage with similar PK findings. We went on to look at other things in spinal fluid with Dr. Musa, and one of the most interesting and most significant differences was this CSF MMP9, which is matrix metalloprotein 9, which is kind of a marker of blood-brain barrier integrity. And it was almost 50% reduction in the resveratrol treated compared to the placebo treated individuals. So we found decreased CSF MMP9, suggesting a neuroprotective effect, and sort of goes along with the greater brain volume loss that we saw with resveratrol compared to placebo. We also looked at the resveratrol reduced the level of plasma pro-inflammatory markers, increased CNS markers of adaptive immunity, MDC, FGF2, and interleukin-4, suggesting long-term benefit immune response. The resveratrol reduces brain volume, suggesting attenuation of CNS edema as a result of the immune modulation. And again, we would like to explore this further with biomarker-proven Alzheimer patients. The initial paper was not biomarker-proven Alzheimer patients, but of course now we can easily confirm the diagnosis with biomarkers. So does anyone have any questions about resveratrol, or anyone have suggestions or discussion?
Well, Scott, I wanted to ask a real quick question, if I may. So if I heard you correctly, you were saying that you started at 500 milligrams per day and went up to 1 gram per day at the end of the trial?
1 gram twice a day at the end of the trial.
2 times a day. And so I guess you didn't do any sort of comparison. You synthesized all of your own resveratrol, so it's not really a comparative study where you compared it against other natural sources of resveratrol.
No, we wanted, we wanted a pure compound. We wanted to know exactly what we were giving patients. So in order to do that, we couldn't use any commercial products. We had to manufacture our own, which was, more than 99.99% pure resveratrol.
Okay, thank you for that. I'll let you continue.
I had one question, if I might. You mentioned that the anti-Alzheimer's effects might be limited to the ApoE4 carriers. Was that exclusive for all the benefits you showed? Did you?
No, it was the CSF A beta 40 was so robust that we also found this in the non-carriers.
Okay.
But most of the significant differences were in the carriers.
Okay.
This is kind of similar to, when you do Alzheimer trials, you usually see a different outcome in the carriers versus the non-carriers. Sometimes it's the other way around.
But I'm wondering, in terms of the anti-cancer effect and any other beneficial effects, were they limited to or greater than non-ApoE carriers?
We didn't look at that. There was only one cancer in the resveratrol group and 6 in the placebo group. So it was significantly different. We didn't look at we didn't look to see if ApoE had an influence on this.
Okay, thank you.
These are only really small numbers, and I'm not sure if you can make any conclusion out of that. But in the meantime, while we're trying to get funding for the next resveratrol trial, I started working with Dr. Musa more closely, who has developed a research program looking at tyrosine kinase inhibitors for neurodegeneration. And one of the drugs that he's looking at is nilotinib, which is also called Ticinia, which is approved for leukemia and has been marketed for, I think, and prescribed for about 12 years now for leukemia. So much is known about this drug and its relative safety, and it's very effective and was a breakthrough in cancer treatment. And the question is, why are we looking at this in neurodegeneration and Alzheimer's disease? And the reason why is that we think it's an ABL inhibitor, but more recently, Dr. Musa thinks its main target is more DDR, which is another class of tyrosine kinases. And I'll let him discuss or refer you to his work as to why he thinks it's more DDR than ABL. But the tyrosine kinase inhibitor is phosphorylate Parkin, regulate Parkin activity, and Becklin, which regulates autophagy. So what we're really interested in is autophagy, and perhaps some effects on inflammation as well with these tyrosine kinase inhibitors. And so we launched this trial of nilotinib. So here we have the autophagosome, and normally the mutant proteins aggregate, whether this is Huntington for Huntington's disease, or alpha-synuclein for Parkinson's disease, or amyloid, or tau for the amyloid and tangles of Alzheimer's disease. So alpha-synuclein for Lewy body dementia, etc. So we think that these autophagy enhancers will help to degrade these neurotoxic aggregates in neurons in the brain, sort of the working hypothesis. And Dr. Musa has done a lot of work with transgenic mouse models, transgenic APP amyloid models, transgenic tau models, transgenic TDP43 models, and transgenic alpha-synuclein models. With mouse models, seems to work very well. And now we're trying to translate this into human studies. And there has been some work already of nilotinib with Parkinson's disease showing some clinical benefits in phase 1 and phase 2. And so we launched this clinical trial of Alzheimer's just to show you some of the results with Alzheimer mice. This is fluorescence of amyloid plaques in a transgenic mouse model treated with either DMSO or nilotinib. So you can see this is the control mouse with all of these amyloid plaques. And here's the treated mouse with much less amyloid plaque. And this is with nilotinib. And here's another tyrosine kinase inhibitor called bosutinib. And quantification of these amyloid plaques is a marked reduction in the amyloid burden of the mouse treated with the tyrosine kinase inhibitor. So we launched our clinical trial. This was a much smaller study. We only ended up with 20 and 17 patients. So a very small phase 2 pilot study to check safety and biomarker outcomes. And then we looked at A beta 40 and 42 in spinal fluid. And there was much less A beta 40 in the treated group compared to the placebo group, as well as CSF A beta 42. So the nilotinib is having effects on CSF A beta levels, as well as their ratio. And these effects are in the favorable direction. Also less phospho-tau 181 in the treated group compared to the placebo. And also slowed hippocampal volume loss by volumetric MRI. This is the placebo group and the nilotinib treated group. So all biomarkers in the favorable direction. But probably the most exciting result from this small phase 2 pilot was the amyloid PET scan. So this is the amyloid burden in a patient treated with a placebo at baseline and then 12 months later. can see an accumulation of amyloid during the course of the study. Compared to the treated group, and this patient has, of course, much less amyloid than this patient, but there's a reduction in amyloid burden by the nilotinib treatment. And when we quantified these results, the only one that was significant, because these were small numbers, is the SUVR temporal lobe, where there was an increase in the placebo and a decrease amyloid burden in the nilotinib treated at 12 months after treatment. And frontal lobe was also significant. Increase in placebo, decreased in nilotinib. As far as the clinical outcomes, this was a study was too small really to even think about anything significant in any clinical outcome, but the trends were in the right direction. For example, the MMSE score, nothing was significant, but the placebo group had a slightly more decline compared to the treated group, and this was found in all the other cognitive and functional outcome measures as well. So none of these are significant, as we would expect from a small phase 2 pilot, but they at least are going in the right direction. And as far as side effects, nilotinib does have a black box warning for arrhythmias and QTc prolongation. And so this is something that is watched for in the leukemia patients. But we used a lower dose than that. We used only about a quarter of the dose that is used for leukemia. And so we didn't run into any EKG abnormalities. The only abnormality that was significant was more mood swings in the treated group compared to the placebo group. And all the other adverse events were no significant difference. And there were, fortunately, there were no serious adverse events in the nilotinib treated compared to the placebo group. So nilotinib is an example of one of these tyrosine kinase inhibitors that we were examining for repurposing. It seems to be safe and well tolerated in older individuals. Reduce CSF phospho-tau, beta-42, CNS amyloid burden, and hippocampal volume loss. And we also think this is a possible disease-modifying treatment.
Sorry.
So we are we're also, can someone please mute their microphone?
Treatment, yeah.
We're also studying nilotinib and other tyrosine kinase
There is
inhibitors.
a large clinical trial.
Zara, I think you need to mute. Sorry to bother you, but we can hear you.
So we now have 3 trials of different tyrosine kinase inhibitors for patients with Lewy body dementia, based on these promising results with Alzheimer's, as well as promising results with Parkinson's disease. And we're trying to gather funding for either another phase 2 or a large phase 3 of nilotinib for Alzheimer's. In the meantime, others are, of course, are interested in this strategy. And here's a paper of a phase 2b/3 randomized double-blind placebo-controlled trial of masitinib, which is another tyrosine kinase inhibitor, which this was a much larger study, larger sample sizes. And so it has had significant benefits in cognitive and functional outcome measures, such as the ADAS-Cog and the ADCS-ADL with masitinib. These are not published yet, but they have been presented publicly. So this is another example of a tyrosine kinase inhibitor by a different group. And this was based on a smaller phase 2 pilot that was published back in 2011, showing a benefit even in the ADAS-Cog in this smaller study with masitinib. In the meantime, Kirkland and his group have been developing quercetin and dasatinib for Alzheimer's disease. Quercetin is another polyphenol, similar to resveratrol, with different structure. And dasatinib is another tyrosine kinase inhibitor, and he's promoting this combination based on a completely different mechanism, and that is called senolytic therapy, which is basically removal of the senescent or dying cells in tissues. Now, this makes sense in other tissues, such as skin or liver that has cells that are turning over and dying, but I'm not sure if this really makes a lot of sense for Alzheimer's disease, which may not have these senescent cells, or maybe it does, because There is some positive findings from Alzheimer mouse models, of course, as usual. Now, senescent cells can cause local and systemic inflammation, tissue destruction, immune system inhibition, and stem and progenital cell dysfunction due to this senescence-associated secretory phenotype, or SASP. So these senescent cells are not only disruptive, but disruptive of the normal cells around them. So if you can develop a strategy of Q plus D for targeting these senescent cells called senolytic therapy. So a clinical trial of this combination is now in progress for Alzheimer's disease. It's a phase 2. We won't have results for several more years. But my eventual goal, once we get more data with resveratrol and nilotinib, is perhaps to do a combination trial of resveratrol and nilotinib, which would have complementary mechanisms of action and perhaps even show greater benefits in the biomarker outcomes and the clinical outcome measures. So that was a lot of information very quickly. So I hope I left time for lots of questions and discussion and comments and suggestions.
Thanks, Scott. That was great. I just have a lot of questions, but I probably only have time to ask one or two, and then of course we'll give others the opportunity to ask some questions. So you had mentioned the blood markers, and I was at the AAIC meeting when they released some of that data about the predictability and accuracy of some of those markers. Would you consider those the standard of care now in the US?
The standard of the best developed biomarkers are the amyloid PET and the CSF proteins. And now more recently, the blood A-beta 42 and 40 is commercially available. These are the ones that have been developed the best so far. However, the first biomarkers that may become abnormal are phospho-tau, for example, phospho-tau 217. These are not quite ready for prime time. These are still in the research stage, and these require highly sensitive methods, Simoa and mass spec technologies, which I don't think are really scalable. So the blood A-beta 42 and 40 is pretty well developed already, but the phospho-tau seems to be the most exciting, but needs a lot more work still.
Okay, just one more quick question. So I guess the pandemic gave everyone an opportunity to develop their web-based screening methods for clinical trials. I'm just wondering, how did yours go?
It's obviously the pandemic really disrupted all of our clinical research and clinical trials. We were on hold for a while. We had to decrease the number of study visits. Fortunately, we didn't have many dropouts of the trial completely. Some of our trials switched to visiting home nurse, for example, to continue the infusions. We had to put a hold on any new clinical trials. So now we have this huge backlog of clinical trials that are just about to get started. And we're still continuing with our screening, and one web-based study that is available to anyone over 50 is called the APT Web Study. So aptwebstudy.org. You go to this website, it gives you information about how to preserve your brain health with aging. And you can also do brain games and check your own cognition. And you can repeat these games every 3 months, 6 months, or whatever you want to do, and track your cognitive performance over time. And this website, if it detects a decline in your performance to the perhaps range of MCI or dementia, will send you an email and saying your cognitive performance is going downhill, and you may want to contact this research site close to you to see if you want to learn about any clinical research opportunities that you might take advantage of. And of course, you don't have to call. It's entirely up to you. But this is sort of another web-based screening method to find the earliest individuals in the AD spectrum.
Okay, so I did want to ask one more question, and then I'll open it up to the floor. So given, you had mentioned caloric restriction, and of course I worked in a lab where that was very popular in Mark Madsen's lab many years ago, but some of the recent work has actually been a little bit more focused on intermittent fasting. And I'm just wondering what your thoughts are on that with regard to stimulating these sirtuins and in a parallel fashion to resveratrol and its effects. And can you comment on that?
Yeah, the older studies was just basic plain caloric restriction. Every day was 2/3 normal calories. But then Dr. Matson has done additional work since then, suggesting that intermittent fasting is even better than this continuous. And perhaps it's this, pulsatile autophagy hypothesis. So the intermittent fasting may be something even better, and maybe we can reproduce this with pulsatile stimulation of autophagy, either with resveratrol or nilotinib. But yeah, the intermittent seems to be even better.
Right, and so I guess, and you already alluded to it, but so it almost appears like resveratrol might be simulating that sort of process. Would you say that's true?
Potentially, yeah. We really don't know what the mechanism is. We haven't tested the mechanism in either one of these clinical trials. We don't really know if we're hitting the target that we think we may be hitting. The sirtuin assays have been tricky and have led to some controversy in the literature. We don't really know what the mechanisms of these, there's a black box between the drug and these Alzheimer biomarker outcomes. And it may or may not be autophagy or sirtuins.
Right. Okay, thank you. So let me open the floor up for questions. I think I saw one or two appear earlier, but while people are formulating questions, I'll ask one more here.
There's one. We have one.
Okay. You can just ask the question yourself, or I can repeat it for you.
Yeah, I have a question. I'm Thomas Nettigarden. I work with resveratrol in heart disease, and it's very much of interest for me to see this exciting presentation with great data from the clinical trials on resveratrol. So my question pertains to the weight loss which you have observed with in your study. Generally, in the previous trials, up to 1 gram is considered safe, and beyond 1 gram, up to 5 grams there are issues. But most of the trials have shown minor GI issues, and this is the first time you're showing weight loss, which is kind of cool because it's not a bad weight loss. I presume it's a good one. So is it because you probably increase the dose from 1 to 2 grams, maybe and not gone all the way to 5 grams, which has been shown to be deleterious?
Yeah. When it came time to picking the dose for resveratrol, because it's metabolized so quickly and levels are so low, I tried to pick the maximal tolerable dose based on the literature. And that came to about 1 gram twice a day, which is what I chose for the trial. And there is this weight loss. There's known weight loss with resveratrol from other studies. It may be because of the GI side effect, but we had a lot of GI side effects in both arms, and it wasn't significantly more, but it's certainly possible. I think there's some mechanism that's maybe not GI related that causing the weight loss, and weight loss in the Alzheimer world, weight loss is not a good thing because the disease causes weight loss, and perhaps the cholinergic medications as well cause even more weight loss. So people are losing weight already. So we don't necessarily want people with Alzheimer's to lose more weight. So that was not particularly a good outcome. But it was certainly apparent, and it was one of the findings that was different. Now, I think if we had gone up beyond 2 grams a day, I think we would have had much more in the way of GI side effects. I think there have been a few studies with 5 grams a day, but those were much shorter term. And when you get to the maximal toxic dose in mice, basically you have to give so much resveratrol that you actually cause GI obstruction as the major toxicity of resveratrol in the mouse.
Yeah, I have had another question regarding the concentrations you showed of resveratrol and its conjugates, the sulfates as well as the glucuronides. There's there's nanomolar concentrations of parent resveratrol molecule, and there's, I think, correct me if I'm wrong, a very high, relatively very high concentration of both the sulfates and the glucuronides. So does it, is it possible that the concentrations of these conjugates might be contributing to the effects you see, given the very low nanomolar concentration found in the blood as well as in the CSF?
It's certainly possible. I tried to find some literature to support that. I think I found, maybe a small amount of literature to support that these metabolites may also have some biologic activity in addition to the native compound. There is certainly much more higher concentration of the metabolites than the native compound in blood and CSF. I don't think it's converted back to the native form in tissues, but certainly these metabolites may have some.
Okay, thank you.
Scott, I also wanted to ask you about natural sources of resveratrol. I mean, it was my impression that we find the most in grape skins. Is that really true?
Yeah, it's in a lot of foods. It's in red grapes, red wine, red grape juice. I recommend the Pinot Noir from Oregon, if you, because this has higher levels of resveratrol. Now, the grape plant obviously makes resveratrol. We can't make our own resveratrol. But when you stress the grape plant, for example, by cold weather or by fungal infection, it increases the levels of resveratrol. And the question is why, and what's the mechanism, and what is it doing? And can we mimic that or take advantage of that by just giving exogenous resveratrol?
Okay, very interesting. And of course, the other interesting aspect to both of your studies is that if I looked at the tables correctly, you found statistical reductions in various cancers in both studies. And this is not always intuitively obvious that one would see benefit in both Alzheimer's and cancer. And I was just wondering if you'd like to comment on that.
Well, cancer is a disease of aging. And is thought to be due to maybe perhaps accumulation of DNA mutations or a breakdown of DNA regulation. So what Alzheimer's and cancer have in common is that they both markedly increase with aging. So, so here's the impaired DNA repair, perhaps leading to more cancers, and then there are other mechanisms. So what they have in common is aging, and if you give a drug that treats aging, there's always this concept of, is aging a disease? Well, maybe, maybe not. There's a trial starting of metformin as a drug that maybe slows aging as a disease. That trial is just getting started. But so because it's targeting aging, you would expect a decrease in cancer and a decrease in neurodegenerative diseases, as well as decrease in everything associated with aging, similar to what we see with these caloric restriction models. So you can look at cancers or neurodegeneration or many other physiologic outcomes. And the caloric restriction has a impact on many outcomes.
In fact, along that line, there's a question in the chat box, and I'll just read it to you. Regarding caloric restriction and its simulation using resveratrol, is it applicable to use in those who have diabetes?
Yes, it is. In our study, I didn't show this because it approached significance, but it wasn't. There was improved glucose tolerance with resveratrol, and so it has been studied. We excluded diabetics from our study, but it has been studied for improvement of glucose tolerance as well. And then in our study, we did find some effect on improved glucose tolerance, but it wasn't significant.
Okay, very interesting. Well, Scott, that was a great talk, really excellent presentation, so much useful data there. And I want to thank you again for your time today. I just want to tell the audience that this talk was recorded. So I'll share that with my social media later today on LinkedIn, Twitter, Facebook, and then on YouTube. So if you can tell your friends who weren't able to come today, they can watch it, the recorded version, if you follow me in any of those social media venues. So thanks again, Scott. Really great to see you. And thank you so much for your time.
Thank you.
Okay. Goodbye, everyone.